Hello all, welcome to our channel Biotech Chat for another video on CSIR NET Life Science. In this video, we will discuss some of the important unit topics and patterns. At this point of time, many of you will have doubts whether you can clear CSIR NET Life Science within 3 months. I can say it is definitely possible to clear NET Life Science within 3 months if you target high weightage units. So total units would be 13. If you target at least 7 to 8 units, you can definitely clear NET Life Science. Some of the high weightage units like unit number 1, 2, 3, 4, 5, either 6 or 7, 11, 12 and 13. If you cover these units along with PYQ and daily practice questions, it is possible for you to get JRF. Definitely. We will discuss some topics which you can study and clear NET. So unit number 1, this unit comprises of all the biochemistry fundamental topics like enzymes, enzyme kinetics and mechanism of enzyme catalysis. In this topic, you can definitely expect questions in a problematic based on majorly on enzyme kinetics and proteins. Proteins have fundamentals of proteins is like a major important role in biochemistry. You have to study Ramachandran plot, secondary structure of proteins, domains, motifs, turns, loops, and you have to study some of the proteins pH. You can expect 2 mark questions from pH of this protein and that protein. Nucleic acid. In this topic, you can expect questions from the types, type A, B and Z DNA and the structure of nucleic acid. You can expect questions like that. Nucleic acid and amino acid biosynthesis. You guys can definitely expect questions in a sentence based manner, like either statement based manner, true or false, whether they would have given a statement and you can expect whether this has to be true or false. You have to study the biosynthetic pathway, what are all the enzymes or proteins involved in the pathway, everything. Bioenergetics, Gibbs free energy, total free energy. Study all the formulas, definitely all the formulas. If you are aware of all the formulas, you can definitely clear the problem based questions that rises from bioenergetics. And next part would be glycolysis, glycogen metabolism, Krebs cycle and etc. In this topic, you can expect questions mostly in a sentence-based manner, or they would have given a diagram. They will be raising some of the enzymes. They'll be asking what enzyme would be put it in this, put it in here or there, everything. And some of the major genetic disorders that rises from the malfunction of the enzyme, you can expect questions like that from this topic. And basis of carbohydrates, lipids, proteins, vitamins, nucleic acid, you can expect match the following from vitamins, and any of the disorders that rises in carbohydrates or lipids. In lipids, you can expect questions from arachidonic acid. Those are all some of the major important topics that you can cover. If you're covering unit 1, go for the PYQ, analyze the pattern, whether they in this topic they have asked in a statement based manner or problem based questions have come from this question. You study according to that. Biochemistry is a very vast topic. If you want to cover it, you have to cover it technically. Unit number 2 is cellular organization. In this unit, cell cycle regulation, checkpoint surveillance mechanism is one of the major important topic, and it is very easy to study as well if you have prior knowledge in cell cycle. It's one of the easiest topic. Study on CDKs, cyclins and everything. Plasma membrane. We are studying plasma membrane ever since 8th standard, secondary, higher school and everything. You have to be thorough in plasma membrane, the structure, types of transport, diffusion, and sometimes it would be paired with signaling like what what kind of molecules can pass through this plasma membrane, what signal it would give rise to. You can expect in a sentence based manner, mainly in a sentence based manner question. And organelle, it's major important topic in unit 2. If you have known the organelle, the function, structure, and what process it is doing, it is a fundamental topic in cells, like in cells. Organelles that are important are nucleus, Golgi bodies, lysosomes, endoplasmic reticulum, mitochondria. Be aware of the structure, pathway, enzymes that are involved in the organelles, and how it is processed, how the enzymes are working, in what manner the enzymes are working. 90% of the question that rises from the organelles would be a statement based question. So, if you know the pathway, you study it, you revise it you revise it in a daily manner, so that the pathway that will be in your mind. If it comes in a statement based manner, you can see, if you are studying all the options in a statement based manner, you will think like, oh my god, all the options are right. But no, that is wrong. Only one or two options would be right. You have to be aware of that. For that, you have to revise it properly. The pathway that should be in your mind. And receptor mediated endocytosis, it's not that major of a topic, but it also plays a role. So be aware of the pathway and enzyme that are involved in the receptor mediated endocytosis. And there comes cytoskeleton, microtubule and microfilament. Mostly the questions that rises from here would be in a basic manner, like what are all the flow, or what are all the things that are involved in microtubules, microfilaments. You can expect questions like that. And the functions, what function it plays and where it plays. For example, some of the microfilament and microtubule plays a role in cell cycle, like kinesin, dynein. So you should be aware which microtubule or microfilament part that plays a role in some other place. Start connecting and study. And then heterochromatin, euchromatin, telomerase, transposons. This in the part this part of this unit, you can connect it with the unit 3. You can study. The question that rises from this topic would be mostly match the following, true or false questions, and how come, which mutations that gives rise, if there is a mutation in this chromosome, what happens. So you can expect questions like this. Connect this topic with unit 3. Unit 3 would be molecular biology. It is one of the easiest unit you could study, and it also has very high weightage. Please don't leave the unit. You can prepare a pattern for this unit and then you can start. The first topic would be DNA replication. Understand the process, understand the pathway, study all the enzymes that are involved in DNA replication and the enzyme, their functions, how it plays the role, where it plays its role, everything. Mostly the question here would be in a sentence based manner, or true or false. They would have given the question and they'll say if the DNA polymerase 1 or 2 that involves in prokaryotic or eukaryotic, true or false. They would be asking question like this. Study the structure of DNA polymerase, types of DNA polymerase. You can expect question, match the following question from here. And in both prokaryotes and eukaryotes, study what are all the enzymes separately involved in both these things. And the major part you can cover here is repair system, DNA repair system. You can connect this topic with the cell cycle regulation, checkpoint regulation. Also, sentence based question would be coming here. Study all these topics. Transcription, same thing. Study the flow, pathway, enzymes that are involved. RNA polymerase plays a role in transcription. Study its types, factors that are involved. You can expect match the following questions from here. And capping and polyadenylation. See, this is very small topic, capping and polyadenylation, but it has so many enzymes involved, and capping also has types involved. Study the pathway clearly on this. You should be clear, you should be thorough with this. Study the structure, how it is structured. And RNA editing process, it also has a role, but it's like a minute topic. You can cover it definitely. And types of RNA and transport. How many types of RNA, how lengthy, where it is produced, how it is produced, what is the flow, what is the purpose for it's to be produced, everything you should study. Operon, regulation of prokaryotic transcription, lacoperon, tripoperon, its process, mutation, everything. Study how mutation plays. If the lacoperon lacks any laci enzyme, laxi enzyme, how it is affected, what happens if it lacks any of the enzymes. Be prepared on that pattern topic. Then translation. tRNA. If you are going to prepare translation, be thorough on tRNA, charging of tRNA with the amino acids, and how many classes, and what is the reaction, structure, types, and function. Amino isolation of the tRNA, that is called charging. Uh, Structure of tRNA. You can also study the degeneracy of genetic code. That is, for example, one amino acid has one or more codons. You should prepare the table properly. Ribosomes and ribosome formation, initiation, how many rRNAs and R proteins are involved in which ribosomes, bacterial ribosomes, mammalian ribosomes, etc., and everything. And post translation modification. You can connect the post translation modification with the chromosome of unit 2 topic. How you can overcome the mutation, and what should I say, how you can modify the DNA, methylation, deamination, and page infection also you can study. What mutation activates or deactivates the DNA modification, whether if there is a mutation in lysine H3, whether it is activating or deactivating the process, you should study. And readers and erasers of post translation modification. You can also study polychrome protein. It is one of the upcoming questions. So nowadays, questions are rising from polychrome protein as well. You can study epigenetic signal. That is a major part you can study. And then you can go for some of the DNA binding domain, zinc finger domain, helix turn, helix motif, helix loop, helix. Study the structure, how it is stabilizing, what are the stabilizing proteins involved, whether it is degrading or it is activating. You can study that kind of pattern. And CRISPR Cas, mRNA silencing, role of chromatin, which you can combine it with the unit 3 itself. And the major part you can study in this unit is what are the antibiotics or any of the enzymes or antibiotics that are inhibiting the process. Some of the antibiotics can inhibit translation process, transcription process. Be thorough with the table. How many antibiotics are there, how it is inhibiting, what is the process it's inhibiting, and study all the factors that are involved in so far process of replication, transcription, translation, the factors and their general functions, whether it is initiating or elongating the everything. Unit number 3 is a short unit, simple unit. You can read it easily. Next we can go for unit number 8, which is another heavy weightage unit. This is also a simple unit if you are from a genetic background or biotech background. Botany students sometimes they can lack this, but if you have studied once this unit, you can cover at least 30 marks from this unit. One of the high weightage unit you can ever study. Questions would be, you can expect direct questions in 2 marks. As far as 4 mark goes, you can expect any problematic questions, pedigree questions, and everything. Some of the important topics include multiple allele, complementation analysis. You can expect a table formed question from complementation analysis. So prepare PYQ on every questions. Genomic imprinting, it is a sentence based questions from this topic, like what are all the disorders that can arise from genomic imprinting. You should prepare penetrance, linkage and crossing over, maternal inheritance. You can expect most of the statement based questions from this topic, so be aware of that. Linkage mapping, tetrad analysis and mapping. You can expect one question from this, that would be an experimental questions. You should know how to calculate mapping unit, how to calculate the distance between two genes, whether it is linked or unlinked. It's an experimental topic, you should study. And the next would be pedigree chart, karyotyping, polygenic inheritance, QTL mapping, etc. You can expect normal, be aware of all of the inheritance, pattern of inheritance, so that you can solve pedigree chart easily. And mutation, what are the type of mutation, causes for the mutation, how to detect the mutation, if it is a single cell mutation or multi cell mutation, how to detect, what you can use to detect the mutation. It is an experimental part. Chromosomal alteration, it's a diagrammatical part. If you understand the diagram from this topic, you can definitely clear this. And be aware of the disorders that can arise from chromosomal alteration in deletion, duplication, inversion, translocation and recombination. Be aware of all the genetic disorders. Study their disorders, why it is arising, where it might give rise to the disorders and everything. Microbial genetics, transformation, transfection, conjugation. You can expect little questions from here. That would be a direct question. You can solve it easily. One of the high weightage unit peoples. So please don't ignore unit 8. And here comes the mother of all units, unit number 5. So far what we have seen, the past 4 units are the fundamentals of biotech or life science. You should be aware of everything that goes on in a cell and the biochemistry background and everything. Those are fundamental. In developmental biology, we will see how it gives rise to an organism and how it gives rise to a plant or anything. You can expect questions from commitment, specification, types of specification, and some of the experimental questions from induction and competence, stem cell, types of stem cell, what stem cells could be used, pluripotent stem cell, totipotents, and everything. Examples for those as well. And mutants and transgenetics in the analysis of development. If you're studying any, okay, we will discuss this later in the end of this unit. Cell surface molecule and sperm egg recognition, that is fertilization in animals. How fertilization happens, what is the step of the fertilization, what are the enzymes that are involved in fertilization, what if there is a mutation in this enzyme, what happens for the fertilization process and everything. You should be aware of the mutation. Most of the experimental question that rises from the mutation, absence of this enzyme, if it is overproduced, what happened. So you can expect questions like that. Embryos and development and double fertilization in plants, you can connect this with unit number 6 and you can study. Zygote formation, cleavage, blastula formation, embryonic field gastrulation, formation of germ layers in animals. Most of the match come from here. Like cleavage, there are types of cleavage, holoblastic cleavage, meroblastic cleavage, so many types of cleavage you can expect. They would have given types one side and what are all the organism that has this cleavage. You should be aware of the examples as well. Gastrulation, invagination, involution, what is the function of this, what it does. You should be aware of that. Most of the match comes from here. Axis and pattern formation in Drosophila, Amphibia and Check. You should study the formation, gene function, experimental questions. You should be aware of the match and everything. Most of the questions, you can expect one question from Drosophila every year. Every year. You should study the gene, maternal effects gene and its proposed function and what happens to it if there is a mutant phenotype, and what are the segmentation pattern in Drosophila, pattern factors affecting the segmentation pattern in Drosophila, everything. early development in C. elegans, vulva formation in C. elegans, islands induction. Statement based question, people, it's one of the very important topic. Just be aware of all the functions. Early development in C. elegans, you can definitely expect one question from C. elegans. Limb development, mostly an experimental based topic. Regeneration and vertebrates and metamorphosis, it's like a simple topic. No experimental question will be asked from here. Mostly you can expect questions in a match the following. Sex determination, you can connect this topic with the genetic part, that is inheritance biology. How sex determination, how they're determining the sex of the organism, and the calculation part, meta female, female, intersex, be aware of the calculation part. Root and shoot development, SAM and RAM. No experimental questions would be asked from here. If there is a question, you can expect from a diagrammatical based question, and they will be giving the gene and what are the function and match the following. You can expect questions from here, but be aware of the process and flow. ABC model, transition of flowering, the same. Oh, you can expect one question from here. It is one of the high weightage unit, people. You can also score simply 30 marks from this unit if you have knowledge on this unit. Mostly this unit covers experimental part, so you should not muck up this unit. You just have to go with the flow. Understand the topic, understand the process, get in the process, and apply the process. Unit number 4. It is a minimal weightage unit. Like you can simply score some 20 to 25 marks here if you know the process. It is also a simple unit, mostly processed unit only. The unit in this unit, immunology part has major role. It's one of the vast unit, but you can expect questions in immunology like types of vaccines, in what disease we can use the which vaccine, T and B cell epitope, innate and adaptive immunity, structure of antibody. You can expect questions from structure of antibody and its function, how antibodies are generated, VDJ, recombination, classes of recombination, MHC molecule, types of MHC molecule, how MHC molecules are connecting with the T cell or B cell, complement system, types of complement system, and you can expect some of the match the following from hypersensitivity and autoimmunity. If you are planning to study autoimmunity, be aware of the diseases that are responsible and why these diseases is arising, reason for the diseases. And if you are going to first of unit 4, hormones and receptors, G-coupled protein receptors, pathways, cell communication, cell signaling pathways, what are the enzymes involved, what if there is a mutation, that is gain of function in the mutation, what happens if there is a loss of function in the mutation, how it affects the signaling later. Be aware of that part, mostly in experimental part. Get in the process, apply the process. That's the major motto. Signaling pathway, you should study RTK, Notch delta signaling, TGF beta signaling, WNT signaling. TGF beta and WNT signaling you can apply in many other places. So be thorough of that process. You study what are the ligands that are involved, how the feedback mechanism works, how it is starting the signaling, everything. And then cell junction, cell adhesion and role of cell adhesion molecules, definitely match the following questions. Gap junction, extracellular matrix, integrins, neurotransmissions, and what are the neurotransmitters that are involved in cell junction and its regulation. You can expect match the following from this unit, or not an experimental part. Cancer genetic, cancer oncogenes, tumor suppressor genes. You should be aware of the table. Study the table, which genes are oncogenes, which genes are tumor suppressor genes. You connect this with cell cycle and study which genes that are involved in cell cycle are oncogenes or tumor suppressor gene, whether they are it's a gain of function gene or a loss of function gene. Also be thorough in the apoptotic pathway. This is also, you can expect either an experimental questions that would be or that would also be a direct questions. You can expect questions from the apoptosis pathway, caspase, and enzymes that are involved, You can expect questions like that, and be aware of the anti-apoptotic proteins, pro-apoptotic members, BH3 only proteins. They might ask in the match the following questions. It is also a major unit. If you are thorough in this unit, you can score some 20 to 25 marks here. Unit number 6. If you are from a botany background, go for unit number 6. If you are from a zoology background, go for unit number 7. If you are from a biotech or life science background, you understand the syllabus, you understand the pattern, and choose the unit which you are comfortable with. For me, unit number 6 would be way easier. It was really easy to understand. You can expect direct questions from unit number 6. Really easy. If you are going for unit number 7, be thorough of all the process. Now we can see unit number 6, plant physiology. Directly go for hormones. You can expect definitely one question from hormones. Biosynthetic pathway, precursor for the hormone, where it is stored, where it is breaking down, and how it is transported, physiological effects and mechanism of action. You can expect a statement based question from plant hormones definitely. And secondary metabolites, the same. How it is synthesized, where it is synthesized, and what is the process, what is the role of secondary metabolites, and what are the compounds that are involved in secondary metabolites. You can go for that one. um, you can connect secondary metabolites with plant biotic and abiotic path factors, because they might ask which secondary metabolite that is responsible for pathogenic resistant or insect resistant, or how it responds to the function. They might ask questions like that. If you are planning, just go for it. Responses of plants to biotic pathogen effectors like virus, bacteria, nematodes. Uh, Study what are the species that are involved in pathogenic effectors and what are the functions and effector. You can also study the effector name. I can post table in the future. You just mock up the table. That would be really easy for you. And you can expect questions from abiotic factors also, which is not that important of a topic. Uh, Light harvesting complex, C2, C3, C4 CAMP pathways. Photosynthetic pathways, it is, you can expect one question from here. It's a known question, direct question they might ask, like which enzymes that are involved, where the enzymes are stored, and what is the role of the enzymes, how it transports, or what mechanism of electron transport, how many electrons are transported, and how many O2 and CO2 are fixed, structure of Rubisco, role of Rubisco, how many secondary metabolites are produced in the photosynthetic pathway. You should also study the structure of all the hormones, that they might ask questions from that also. And I guess that would be enough in most of the photosynthetic pathways. Be prepared for the genes, proteins that are involved, and flowchart of Z scheme, PS1, PS2, and structure, and also aware of the inhibitors that might inhibit the C2, C3 pathways, stromal opening, etc. Then nitrogen metabolism, you can expect one direct questions from here if they ask. It's an easy part. You can go for night assimilation part. What are the genes that are involved? Which biosynthetic pathway? Which compound are produced? Sometimes they might ask the reaction that are catalyzed by nitrogenase. Mostly, you can expect questions from here. If and all they are asking questions here, they might go for the some of the examples of organisms that carry out nitrogen fixation, like symbiotic nitrogen fixation, for anaerobic nitrogen fixation. They might ask with the examples. So study what examples, which host plant is using which nitrogen fixation. You can go for that. I'll post table for that as well in the future. And then structure, function, and mechanism of action of phytochromes, cryptochromes, phototropins, photoperiodism, and a biological clock. This is also one of the easiest topic you can study. Direct questions they will ask from this part. And then unit 11. It's a small unit, really small unit. I know most of the biotech and life science background peoples, they'll be studying, most of the peoples will be studying ecology and evolution for the first time. Ecology is a very vast unit. So many examples, so many historical events and everything. But if you've chosen evolution, there are few examples, few major events, few historical events you can study easily. Be aware of the evolutionary timescale, major events, scientists' name, and what theories they have proposed, whether there is a graph based theories or anything. Be aware of that. Molecular tools and phylogeny, terms and phylogenic trees. Be aware of the terms that are involved in the phylogenic mechanisms. They might ask in the match the following questions or in a 2 mark questions, and phylogenetic tree cladogram, which they might ask questions like at which point there is a divergent, like that. Hardy-Weinberg equation, it's a problem and concept questions. uh, be thorough the formula, practice pyq and Hardy-Weinberg equation, and practice some of the free questions you can search in the Google. That would be more than enough. Other than that, these two are some of the topics which you can expect in a 2 mark questions. Not that much of an important. Major unit 12 plus 13. If you're starting to prepare unit 13, start with unit 12. Combine both the units, study the units in a combined manner. Unit number 12 has agrobacteria mediated gene transfer. Study the process, study the vector that are involved, study the resistance gene that are involved, function of vir genes, how it is doing, how the process of agrobacterium gene transfer happens, where it happens. There might be a sentence based questions, or you can expect match the following from vir gene functions. Transgenic plants and animals, study all the names of the transgenic plants and animals, where it is discovered, who discovered, how it was discovered, and etc. Molecular markers, it is an experimental topic. RAPD, RFLP, study this along with the PCR question, so you might find it very easy. AFLP, SSR, they would ask question by combining this with Western blot, Northern blot, any blotting mechanisms, and immunotechniques as well. We'll practice some of the experimental questions from molecular markers in the future. And vectors and enzymes. What are the type of vectors? How many types of restriction enzymes? And how we can use it, where we can use it, we can expect some of the experimental questions here. Electrophoresis, hydrogel electrophoresis, SDS page, native SDS page, isoelectric focusing, types, what are the uses, what are the applications, what type of agros we are using in which one. You just study the whole process, basic process, and try to apply it in the experimental manner. Immunotechniques, same, study the whole process, how it is working, what is the function of this, and try to apply it in the experimental manner. If you are getting questions from immunotechniques, just recall your process, recall what is the process of isoelectric focusing, how we can apply it here, what would happen. Biophysical method, UV spec, mass spec, X-ray. Please aware of the wavelength, which has higher wavelength, which has higher energy. Practice PYQ in the biophysical method, and some of the experimental questions they might ask from circular dichromosome, and NMR based questions, etc. Chromatographic techniques, mostly like from HPLC, liquid chromatography, or affinity chromatography, you can expect match from there. Not much of experimental questions, but you can expect match the following from this part of the topic. Microscopic technique, study all the basic terms of the microscopic techniques. What are the staining techniques, fixation, SEM, TEM, fluorescent microscope, you can expect match the following from this topic. ECG, EKG, PET, MRI. Uh, just form a table for this. There will be a terms in each topic. ECG you have terms, for EKG also you have separate terms. Please be aware of the terms, where it is happening, how it is happening. You can expect match the following from here. I will also post the table form of questions in the channel future. Radio labeling techniques, it's not much of an important, but you might see questions connecting through sequencing. If you are labeling one molecule of phosphorus in the DNA, what happens after 3 to 4 steps? You can expect questions like that in experimental based. Study the types of radio labeling techniques, how many types, and what radio labeled item you can use. You can also study sequencing technique. I'm sorry, I forgot to put it in the table, but be aware of the sequencing technique, how it is processed, how it is used, enzymes that are involved in sequencing, that are needed in sequencing. PCR, how many types of PCR, which PCR has how many primers, where it can be used, everything, etc. I'll be posting experimental related questions from now on regularly on our channel in the morning session. So please stay tuned in this channel. We can solve